Have you ever really thought about how scientists can edit the very code of life? I mean, not just read it, but actually rewrite it. It sounds like science fiction, right? Well, today we're going to build the ultimate toolkit for genetic engineering, and we'll see exactly how it works. So every living thing, from the smallest bacterium all the way up to us, is built from the instructions in its DNA. You can think of it as the ultimate blueprint. But here's the thing. This blueprint is just massive, incredibly long and unbelievably complex. To give you some perspective, a single typical chromosome in your body can have over 5 million base pairs. That's 5 million of the individual letters that make up the genetic code. Imagine trying to find one specific sentence in a book that has millions upon millions of letters. It's a huge challenge. Right, that's the big question, isn't it? If you want to study a gene or use it for something, you can't just grab the whole chromosome. You need a way to find the exact start and end of that one little gene and just snip it out with absolute precision. How on earth do you do that? Well, the answer, amazingly, didn't come from some brilliant invention in a lab. It actually came from nature itself. Turns out, bacteria figured this out ages ago, and they had the perfect little tool for the job. So let me introduce you to the first and maybe most important tool in our kit, the restriction enzyme. Honestly, the best way to picture this is as a pair of tiny, programmable scissors. But instead of cutting paper, they cut DNA. And here's the cool part. They don't just cut randomly. They're incredibly specific. So where did we even find these things? Well, they're part of a bacterium's immune system. When a virus, called a phage, attacks, it injects its DNA into the bacterium to try and take over. The bacterium fights back with these restriction enzymes, which hunt down the foreign DNA and just chop it to pieces. It's basically microscopic warfare. But hold on. How does the bacterium stop its own DNA from getting shredded? This is where it gets really smart. The bacterium puts these tiny chemical tags on its own DNA, right at the spots where the scissors would cut. This process, called methylation, works like a little do not cut sign, making its own blueprint totally invisible to its own defense system. And the precision here is just mind-blowing. Each type of restriction enzyme looks for one and only one specific sequence of DNA letters. So, for example, the famous enzyme EcoRI will scan millions of base pairs, but it will only, and I mean only, make a cut when it finds this exact sequence, GAATTC. Anything else, and it just keeps on moving. You can see it right here. The enzyme latches onto the DNA, slides along the strand until it finds that one specific recognition site, and then snip, it makes a clean cut. This is how we can take that gigantic chromosome and slice it into perfectly manageable, gene-sized pieces. Okay, so we've got our scissors, but it turns out that how you cut the DNA is just as important as where you cut. There are two main ways these enzymes can make a cut, and one of them is way, way more useful for what we're trying to do. Some enzymes make what's called a blunt cut. They slice straight through both strands of DNA, creating a flat end. It's neat, but it's actually pretty hard to join another piece of DNA to it. But other enzymes make a sticky cut. It's staggered, which leaves this little single-stranded overhang. And that little overhang, well, that's the secret sauce. You can really see the difference here. On the left, the blunt cut is perfectly flat. But look at the sticky end on the right from our old friend EcoRI. It's got that little tail, that overhang. And that little bit of single-stranded DNA is actively looking for a partner to pair up with. So why does this matter so much? Because these sticky ends are, well, they're sticky. The exposed letters on the overhang want to find their matching partners on another piece of DNA that was cut with the very same enzyme. They work just like molecular Velcro, holding two different pieces of DNA together just long enough for us to make the connection permanent. It makes everything so much easier. All right, we've totally mastered the cutting part, but that's only half the story. Now we need to paste, and for that, we need to pull another amazing tool out of nature's toolkit, a molecular glue. This next tool is called DNA ligase. In nature, its job is to go around repairing breaks in the DNA backbone. But in the lab, we can hijack it to permanently stitch together two different pieces of DNA, basically sealing the deal on our little cut and paste project. So after the sticky nicks have done their job and brought our two DNA pieces together, DNA ligase comes in and makes it official. It rebuilds the final chemical bonds in the DNA backbone, turning two separate fragments into one seamless, brand new molecule. So, we have our molecular scissors and our molecular glue. Now let's put it all together and see the whole gene cloning process in action, from start to finish. Step 1. We take the DNA from our source organism. Let's say we want the gene for human insulin, the protein that helps millions of people with diabetes. We use our chosen restriction enzyme to make super precise cuts on either side of that gene, lifting it right out of the chromosome. Step 2. We need a way to carry this gene. For that, we use something called a plasmid. It's a little circular piece of DNA from a bacterium. We use it as a vector, or a delivery vehicle. And here's the crucial part. We cut this plasmid open with the exact same restriction enzyme. That's how we get those perfectly matching sticky ends. And now for step 3, the final assembly. We mix our isolated gene with all those cut open vectors. The sticky ends find each other, that molecular Velcro snaps into place, and then we add our molecular glue, DNA ligase, to make that connection permanent. And there you have it. The final product is a single custom-built DNA molecule made from two totally different sources. This has a name. It's called recombinant DNA. And this is the absolute cornerstone of modern biotechnology. It's how we make things like insulin and bacteria and create crops with new abilities. It all starts right here. So we started with this massive problem of scale and ended up with a solution of unbelievable precision, all by borrowing tools that nature perfected over billions of years. It really makes you stop and think. With a toolkit this powerful, with the ability to cut, paste, and rewrite the code of life itself, what blueprints are we going to write next?